Hey everybody, what's going on? This week, well, I guess this lecture, since it's a couple of weeks behind-ish, right? We're going to be talking about donuts. We're going to be talking about macronutrients. Before we really go too much further, something we need to talk about, or at least cover, is kind of what is an atom. The reason I want you guys focused on this is simply because it helps you to understand kind of what's going on with the difference between carbohydrates and fats, right? So what an atom is, it is simply the smallest unit of matter that forms a chemical element, right? So we can have a hydrogen atom. It is a proton. It is essentially just a single positive. We're going all the way back to periodic table of elements here. I don't need you guys to memorize most of this. Just have a general understanding of what's going on with it. In general, the human body is going to be comprised of really four predominant different types of atoms, different types of elements. Nitrogen, hydrogen, carbon, and oxygen. Truth be told, the reason that organic chemistry is called such, it's because it's got the study of carbon associated with it. So realistically, any carbon matter containing atom is technically organic. News you can use, right? When we have bonding between atoms and elements or whatever, it's technically just like a union between these elements. There's a couple different types of bonds. For the most part, what we're going to be focused on is covalent bonding, or kind of a chemical attraction or a sharing of bonds. So the reason that glucose is because it's C6H12O6. So that single molecule actually has 6 carbons that are bonded to each other. It's also got 12 hydrogens that are bonded to that, and it's got 6 oxygens that are going to be bonded to that. Realistically, an ionic bond is going to have a positive and a negative. It's going to have poles. It's going to be ionic, right? Ion, positive, negative charge. Whew. Throw back to that high school science, right? Some of you guys are luckier if you've had biology or chemistry more recently. Realistically, big picture thing here, covalent bonds are going to be sharing atoms. So they're fairly easy to break, but they're also got some rigidity, some strength to them. The reason I bring that all up is because of the importance of bonds within carbon. So these carbon bonds, I have it listed as carbon bonds, it's actually covalent bonds, are going to be that shared electron. So if we look at that top molecule, top right, we've got 2 hydrogens and a single oxygen. The big thing here is we almost always are going to want 8 electrons inside of our valence shell. That valence shell is going to be denoted by how many dots we have. So you see here on oxygen, 1, 2, 3, 4. Each one of these lines represents 2 electrons because it's being shared, right? 5, 6, 7, 8. This molecule is water. It's H2O. Down one more row, we're going to have carbon. You can see that carbon has 4 different electrons. That means it can have 4 different bonds. So we could technically attach 4 different carbons or 4 different hydrogens to this. In the image to the left, or to the right, oh my gosh, I'm an idiot. In the image to the right, we're going to have CO2, carbon dioxide, or that product that is formed when we go from pyruvate to acetyl-CoA for all my ex-fizzers. If you look, let's count these really fast. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Right? CO2. Cool, right? Like it kind of makes sense. Let's go down one more. This is a nitrogen. We're going to have on that oxygen 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7. So we technically could add one more unit here. We could add a hydrogen. We could add whatever. This is a nitrogen containing group. Pay attention to that because we're going to be talking about amino acids. That amino means nitrogen. Just a little bit more for you. Let's pay attention to kind of what's going on with these carbons and these hydrogens and what's bonded to what. It'll help you guys remember the difference between the macronutrients. On the far left, if you look, every single time we have one of these like little dip looking things, right? It's going to actually be a carbon. So we have a carbon here. This little point right here is a carbon, 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 carbon. We also have a carbon up here. You can see that we have oxygen, oxygen, CH3, nitrogen, 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 right? So that means it's going to be an amino acid Anytime you see nitrogen inside these components, it's going to be an amine. It's going to be an amino acid. Nitrogen is the denominator. It has to be there for it to be a protein. If we go over 1, we go to the center, we're going to see glucose, C6H12O6. Now look here. We have 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. It might be a carbon copy of each other. I can't believe I just did that. Anyways, we're going to also have 1 oxygen, 2 oxygen, 3 oxygen, 4 oxygen, 5 oxygen, 6 oxygen. Interesting. C6H12O6. Let's count the hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. C6H12O6. Sugar's almost always going to follow that pattern. C6H12O6, CnH2nOn, C12H24O12, right? That carbon is going to be bound to 2 other carbons, a hydrogen, and a hydroxyl group, or an OH. If we go over 1 more, we go to that fat molecule. What is the big difference here between an amino acid and a fat? It lacks an amine. It lacks a nitrogen. What's the big difference between a glucose and a fat, though? The hydrogen to carbon ratio. Look how many more hydrogens there are. Look how many more hydrogens are actually attached to carbons. And if you notice, we only have 2 oxygens in that whole molecule. So the major difference here is that structurally, a carbohydrate has a lower ratio of carbon to hydrogen than fat does. Fat maximizes how many hydrogens we can put on to those carbon chains. Kind of neat, right? And it will become super important later on when we start talking about fatty acid carriers. We start talking about hydrogen carriers and electron carriers when we get into bioenergetics. So why do we care, right? Like, dude, why are you sitting here telling me about carbons and nitrogens and I don't give a flying blah, 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 right? That's what that word means. Well, it's essentially how we're going to be generating our ATP, right? Sneak preview to 2 weeks from now, 3 weeks from now, whenever we start talking about bioenergetics, we're going to be talking about how we can basically strip down these carbons, pull carbons, split carbons, move carbons around to actually make ATP. Same thing with those hydrogens. We're going to be stripping those hydrogens off of those carbon skeletons, putting them onto NAD or FAD, and actually taking those over to the electron transport chain. And that's how we're going to get the bulk majority of our ATP. ATP is the most important thing that exists within a human. If we do not have ATP, we die. We have to have it. It fuels muscle contraction. Without muscle contraction, our heart doesn't beat, our diaphragm doesn't contract. We don't move blood, we don't breathe, we die, right? Super, super important. In general, the big thing with nutrition is how are we getting that sustenance into our body? How do we get the goods into our body? How do we get the prequel into our body, right? How do we get those foodstuffs into our body to actually make energy? Difference between exphys and nutrition, pretty small realistically, right? Nutrition is going to be how we feed our body. Exphys is just going to be how we use that energy, how we adapt to stress with that energy. Realistically, not a huge difference, right? One of them is just fueling us. The other one is using that fuel. They use the same crossovers. They use the same different. You're an idiot, Parker. Right? They use the same processes here. So what are carbs? We already talked about this, right? Essentially, carbs are things that have a lower carbon to hydrogen ratio than fat. They don't have an amine group, so they're following that C6H12O6 or that CnH2nOn type thing. Carbs are going to be divided into one of 3 main groups. We're going to have monosaccharides. Saccharide means sugar. Disaccharides, which means 2 sugars, 2 shug, right? Monosaccharide, 1 shug. I guess 1 shug, just shug. Disaccharide, 2 shug. There we go. And a polysaccharide, shug. So it's just going to be a lot of sugars that are going to be strung together. Polysaccharide, anything more than 2. Disaccharide, 2 sugars strung together. Monosaccharide, un shug. So what are a couple of the monosaccharides? Major ones, really there's 3 primary that are going to be found in nature. We're going to have glucose, fructose, and galactose. I don't care if you call it I don't care if you call it fructose. I don't care if you call it fructose, or fructose or however you want to say it. It all means the same thing. When I was doing my PhD and even during my master's, I had the chance to study under some folks that every single individual called it something different, right? Some of the individuals that were international called it fructose, and that's kind of what I ended up have taken. But you'll hear me jump back and forth between those. Glucose is just dextrose. If you've ever taken high dextrose powder like waxy maize, dextrose powder, this is going to be the most common form of glucose. The other common form of glucose is actually going to be what's circulating inside of our blood. I'm supposed to be showing you my veins by doing that. You probably didn't capture that. So it's going to be the most common monosaccharide inside of our body. It is naturally occurring in foodstuffs. It's already there. It is essentially this massive digestion product. If you eat a potato, we're going to break it down all the way into monosaccharides. A large proportion of that's going to be glucose. So we're going to actually take and break everything down as much as we can into glucose. It's something that we can produce in the body, and something that I definitely want to make sure that we highlight here is that all of the carbohydrate that our body needs can be synthesized inside of the body. There is zero obligate need for carbohydrate in the diet. We can make it if we need it. So there's no reason to have 150 grams of carbohydrates a day for central nervous system functioning. That's not true. It's not backed by science. Don't perpetuate the myth, right? There's no need for carbohydrate in the diet for most healthy individuals. Just add that clarifier in there. And generally, carbohydrate's going to serve one of three sources when we put it into our body. I'm going to use it for energy. We're going to save it for later energy, which would be glycogen, or we're going to convert it to fat, so we're going to store it for longer term utilization. So glucose, right? Inside the blood, transported inside the blood, one of three primary functions: energy, stored energy, converted into a different form for more stored energy. Makes sense? So our disaccharides are just going to be two monosaccharides, right? Remember, shug, di-shug. It's going to be two sugars. So we can actually combine a couple of those monosaccharides to get a couple different types of disaccharides. In general, the three most common and most important that you're going to run into are going to be sucrose, lactose, and maltose. Sucrose, most common form of disaccharide. Think table sugar. It's a 50-50 blend of glucose and fructose. So one of the shug, the single shug, is glucose. The other shug is fructose, right? It can be about 25% of the American diet. Shocking, right? Sugar shocking. Lactose is going to be found in milk naturally. It's a combination of glucose and galactose. So think this is the sweetness in milk that you get, or this is found in cheese. It's the somewhat sweetness that you get in cheese. I think it's also in like smelly feet cheese, right? Like those weird cheeses that you sometimes buy at like Whole Foods or Whole Paycheck. Right? Lactose. Maltose. This is going to be two different glucose molecules. So combining two different glucose molecules. I mean, most important to probably most of us is going to be, this is what's going to be the primary sugar found in beer. Funny enough, if you've never brewed beer, when you actually take and you put maltose and you start kind of dealing with the yeast, all that's happening is the yeast is chewing up the maltose and pooping out CO2 to give you that carbonation and alcohol, right? The good stuff. Here's literally 74 other names for sugar. Just different names for monosaccharides, golden saccharides. I read golden syrup. I don't even know what that is. It's a little alarming. It's just a couple of different names for different types of sugars that you'll see as ingredients in food. So be aware, right? Know some of these. Most of you guys probably know most of them. Agave juice, agave nectar. Anytime you see the word syrup, it's going to be sugar, right? There's a reason why sugar is a punch in the face of freaking sugar, of sweetness. Icing sugar, inverted sugar, duh, it's sugar. It says it in it. Sorghum syrup, high fructose corn syrup, all these different things, just different names for sweeteners, different names for sugar, right? Polysaccharides. This is where things get a little bit bigger, right? So a polysaccharide is going to be 10 plus saccharides linked together, 10 plus monosaccharides linked. So this could be glucose. I think that was 10. It could be whatever. Essentially, this is going to be a storage form of saccharides. It's going to be a storage form of sugar, whether that's in plants or that's in animals. If it's in plants, this is going to be the starch in the fiber. Think about what it is that makes a potato starchy. If you bite into a French fry and you kind of get that distinct starch, that is actually polysaccharide. It's also going to be our fiber. We'll get into fiber in a little bit. It might be the next slide. But know that you can find polysaccharide in plants. In animals, we get the storage form of carbohydrate. Just like we store carbohydrate in human muscle tissue and in the liver as glycogen, which is just a highly branched glucose, highly branched carbohydrate, animals store it the same way. Humans are animals, right? So when you bite into your chicken thigh or you bite into your chicken calf, chicken wing, whatever you want to call it, you're actually eating some carbohydrate. Small amounts, but you are getting some because that animal had stored carbohydrate in it before it died. So let's kind of take and dive a little bit further. What exactly is starch? Well, it's a storage form of carbohydrates in plants specifically, right? You don't ever hear about starchy mussels, but we hear about starchy plants, starchy foods such as breads and cereals, spaghettis, beans, rice, pasta, right? You name it. When you think about complex carbs, right? Like the euphemism complex carbohydrates, close your eyes and picture what you're going to picture for a complex carb, right? You're probably thinking a sweet potato. At least that's what I think of, or brown rice or whatever. That is going to still be starch. It's starch, right? It is carbohydrate that you're thinking of. Realistically, there's two primary forms of starch. We're going to have amylose as well as amylopectin. So we'll talk about those as we get into later lectures about kind of what the difference is with amylose and amylopectin. Fiber, on the other side, is a really interesting polysaccharide. Essentially, humans lack the enzymes needed to break fiber down. So we can't get the goodness out of fiber realistically. But what it does do is it can ferment inside of our gut. We'll talk about that when we talk about digestion. But it can be fermented inside of our gut. There's a reason I mentioned beer fermentation. The same thing is going to be happening here with fiber, right? Our body can actually ferment fiber inside of it. Inside of it, we can provide some sort of sustenance off of this. It's a really cool adaptation that humans have. When it ferments, it creates something called a butyrate. This butyrate is actually a short-chain fatty acid, and it can actually provide energy and nutrition to colonocytes or colon cells. It is exclusively found in plants unless it is synthetically derived. When we get it inside of our bodies, what does fiber do? What do you know fiber being so good at? Right? Making you poop, right? What it's going to do is it's actually going to provide bulk to your stool. It's going to actually make it so you're not just pooping water in some way, shape, or form, right? It's adding, grossly, it's adding shape and structure to your poo. There's a couple different ways that it's going to do that. One of the primary ways is that fiber is a little bit scratchy, and as it's going down through your colon, it's going to actually be irritating the cell linings and the walls inside of the colon, kind of causing that feeling of having to take a dump. Well, think about what happens if you have a low fiber diet. If you're holding a bunch of stuff that our body didn't want to absorb the first go around, how likely is it that it's going to provide something just incredibly useful for us if it doesn't come out? Well, fiber has been linked with significantly lower occurrences of obesity, insulin resistance, diabetes, inflammation, hyperlipidemia, name it, you go on and on and on. Fiber can be extremely useful inside of the diet, despite the fact that we're not pulling nutrition out of it, but it helps us to ferment and provide nutrition to our colonocytes, as well as it helps provide an environment of really good healthy bacteria inside of our gut and helps us eliminate and excrete waste. There's kind of two different types of fiber that have proposed mechanisms for this. Water soluble and water insoluble. Obviously one dissolves in water and one does not. Now let's get into animals because I mean personally I love eating meat. I would much rather eat meat than freaking salad and rabbit food. So let's talk about animals. Glycogen is the primary storage form of carbohydrate inside of an animal. So we're going to store it in our liver and we're going to store it inside of our muscle. Realistically, we have about 500 grams of glycogen stored. We have about 100-ish grams of glycogen stored inside the liver and about 400 grams stored inside of the muscle. When you get to XPhys, if you haven't had it yet, or if you're in XPhys now, this is where we get a lot of glucose for energetic demands. This is why in the textbook we talk about how we don't have to have the initial energy investment of an ATP to become glucose 6-phosphate. Glycogen is already ready to go. So it's actually a little bit more of a preferred fuel substrate for our muscles for burning fuel. Sorry. I need more coffee, I guess, or a Bang. If anyone wants to bring a Bang energy drink, favorite student. Starch, if you look on the left, well, on the right-hand side, but on the left, right, you can see that it's a pretty branched molecule. Go one step over and look at how branched glycogen is. Funny enough, glycogen actually pulls 3 grams of water per gram of it. So if you eat a ton of carbs, it's the reason why you feel like you're holding water, because you are. You're literally puffed up from glycogen as well as from water. Same reason why when you cut carbs within the first couple of days, you actually lose a pretty dramatic amount of weight, because every gram of glycogen that you lose because you're not eating carbs, you actually lose 3 grams of water. Kind of a neat little trick. It's actually what a lot of people do whenever they're making weight cuts, is they kill their glycogen and pee off most of it. They're not just losing tons and tons of fat. Kind of neat. For every 1 gram of carbohydrate, this is important. Sorry, I didn't mention it earlier. For every 1 gram of carbohydrate, you get 4 calories of energy, right? Every 1 gram of carbohydrate equals 4 kcals of energy. This slide will make a little bit more sense when we start talking about bioenergetics next week, but in general, well, I guess it's in 2 weeks. Regardless, this slide will make much more sense in a couple of weeks. Essentially, glycogen can be one of our major sources of fuel. When it comes to carbohydrates, we have storage form in the blood sugar. We have our liver, and we have our muscle glycogen. At all costs, we want to try to hold on to a certain level of blood sugar. If our blood sugar dips too low and we're not ready for it, it can result in really bad things. Our brain can lose energy. If our brain loses energy, we get CNS dysfunction, and we generally don't feel good, if not end up in a coma, if not die, right? So we have to protect our blood sugar in some way, shape, or form. So if we need carbs, we can actually go and burn our liver and our muscle glycogen. Our liver glycogen kind of gets broken down into sugar, into glucose, and gets released into the blood to increase the blood sugar and become available for muscle if it needs it. Muscle glycogen is actually going to serve as kind of like a local energy source. So it's ready to go. It'd be like if this was a Bang and not water, I'd be a lot happier, but I'd have energy source right here ready to go. I wouldn't have to walk down the stairs and into the kitchen to grab an energy drink. Does that make sense? In general, there's 2 primary hormones that are going to really control how glucose functions in the body. There's significantly more, but we're going to focus on 2. Let's focus on insulin and glucagon. Insulin is released from the beta cells of the pancreas, and it's going to result in increased uptake of glucose. It's going to draw glucose into the cells. It'll bind with a receptor. That cell will open up and bring in glucose. So it's going to lower blood sugar. That's its goal. Glucagon is the complete opposite. Glucagon is going to be responsible for helping to increase blood sugar. So think glucose is gone. So when glucose is gone, glucagon gets released. So we want to increase the blood sugar. We've kind of already hit on this, and I don't want to kind of berate you guys with how important carbohydrates are in the body, but in general, we're going to use them for ATP, right? We're plugging them into glycolysis. Glycolysis can move into Krebs. Krebs can move into ETC. We can make energy. But what's also really cool about carbohydrates, and is important to know how to manipulate when it comes to things like weight loss, is that it's going to be responsible for controlling the fasting and fed status. When we eat carbs, our body automatically switches over and actually switches into a fat storage zone. We switch from a fat breakdown and fat burning zone into a fat storage zone. So we switch from anabolism, or from catabolism to anabolism. So we're actually switching. Glucose in the diet is going to increase insulin. That insulin is actually going to signal the switch from a fasting to a fed state. So it's going to help spare nitrogen. It's going to help spare muscle protein from being broken down, but it's also going to prevent our ability to burn fat. Super important for later in the semester. Carbohydrates increase insulin. Insulin blocks fat burning. All right, gang. So let's keep on moving. Let's talk about lipids. Lipids, as you guys can probably guess by what's on the background, it's just going to be a form of fat, right? So if you think fatty meats, cheese, butter, whatever, it's going to be lipid, right? Lipid is fat, fat is lipid. Word means the same thing. If you're like me, then when you see an avocado, you sit there and think, but I'm the healthy kind of fat, right? Like, it's fat. I don't know how else to cut it. Lipid sources are really neat, right? And something that I really enjoy personally. So it might come across that way. If it does, I apologize. Fat's just a really fascinating food source. So fats are going to be comprised of oils, fats, waxes. There's so many different ways that you can eat fat. Think about it, right? Like when you eat meat, it's going to be a little bit more solid. If you're eating olive oil, it's going to be a little bit more liquid. If you eat something like coconut oil, depending on the temperature you're at, it could be frozen or it could be a liquid. It's kind of a really cool substrate. I don't want you guys to get too caught up in this, but I want you to know that carbohydrates and fats are going to be predominantly different based on their hydrogen to carbon ratio. And the reason that becomes important is for things like saturation. We can actually stack more fats in a row in parallel than we can with carbohydrates because of the way that it doesn't necessarily branch. Like carbohydrates kind of branch all the way through. We can actually store fats kind of stacked. So it helps us kind of create this saturation, if you will. We'll talk about it, but it's kind of a cool aspect to it. Similarly, kind of like we can get carbs from both plant and animals, we can actually get substantial amounts of fats from plants and animals. You don't actually have to eat meat or eat fish in order to get fat. You can get almost all of it exclusively from plants. Is it more difficult? Yeah, of course, right? Like anytime you're not going to eat meat, it's going to make your life a little bit more difficult, mainly because it's not going to taste as good. But two, it is going to be sizable in terms of the difference in fat availability and fat quantity. Just like we have with carbs, we're going to have simple fats, we're going to have compound fats, and we're also going to have something called derived lipids or derived fats. And we'll kind of talk about each one of those. One of the terms that I'm sure you're familiar with is saturated versus unsaturated fat. This is where that hydrogen and carbon bond kind of becomes important, right? So up here on the top right, you're going to have a saturated fat, and we're going to have carb, blah, 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 right? And each one of those is going to be attached to a certain number of hydrogens. If you go down and you actually look at that unsaturated fat, you'll see that there's actually a double bond. That double bond causes a kinking because there's no hydrogens here. So because there's no hydrogens down below and we have that double bond, it's actually going to force the second half of this carbon string to kind of move towards that empty space. So if we were to stack another line and another line and another line, it would not be as saturated, as densely populated with carbon and hydrogen as it would be if it was this straight line. So it's all the differences, right? Saturated fat, we can essentially add a bunch in parallel. Unsaturated fat, we can't fit as many in a per unit basis. So we kind of end up having a difference in structure at different temperatures, and we'll talk about it. So unsaturated fat has double bonds. If it has multiple double bonds, it's polyunsaturated. Anytime it's two or more double bonds, polyunsaturated. One double bond, monounsaturated. Most of these unsaturated fats are going to come from plant sources, whereas most of our saturated fats are going to come from animal products. Another kind of primary fat type is going to be trans fat. If you look, we have saturated and we have unsaturated. Think trans fat is kind of like somewhere in between those, essentially. This is going to be a marvel that happens whenever we mess with the fat, basically. We're trying to create a saturated fat structure-ish and characteristic by using unsaturated fats. So again, carbon-hydrogen chain, it's going to kind of stack nice and neat. If we look at this double bond here and this double bond here, we have a polyunsaturated fatty acid because we have two double bonds. If we then look at this trans fat, we don't have that same level of kinking. We have the kink, but it doesn't kind of about face and change direction completely. It ends up going on to the other side. And you can see it's because we've actually taken and put this hydrogen here, and we've moved this hydrogen over to the other side. So if you look over here, you can see it's a cis double bond, so it's the same side hydrogen. Hydrogen are on the same side of the chain. Down here with this trans fat, it's actually on opposite sides of the chain. So we can create a saturated fat characteristic, such as margarine, which is spreadable and somewhat soft at room temperature, and we can kind of create that with an unsaturated fat that would normally be in olive oil. It's kind of a neat process. Now, omega fatty acids, these are going to be just how we describe where that double bond is. So we go to the terminus end, that far end, and we say, okay, how many bonds in is that double bond? How many carbons in is that double bond? For this one, it's 1, 2, 3 carbons in, so it's an omega-3 fatty acid. This one here, omega 1, 2, 3, 4, 5, 6. So it's an omega-6 fatty acid. We also have omega-9 fatty acids. What's kind of neat about omega fatty acids is that they are essential. Whereas we have really no essential carbohydrates, we actually have essential fats. We have to consume them in some level in order to live and exist because our bodies can't actually make them. Whereas we can make carbohydrates, we actually can't make certain types of fats. Just like we can't make certain types of protein, we have to consume them. So where are we going to get these? Well, we can get omega-3 fatty acids from cold water fish, shellfish, that type of stuff. Omega-6 is going to be our nuts and our seeds and our vegetable oils. And then omega-9 fatty acids we can get from olive oils, almond oils, walnuts. So those fattier nuts, as well as some animal fat as well. When you start looking at grass-fed beef versus grain-fed beef or grass-fed pork versus grain-fed pork, this is where those things kind of start to come out, because we can actually change the fatty acid structure of an animal, of ourselves or an animal, by manipulating what it eats in the diet. So if it eats a heavy grain diet, it's going to be higher in certain types of fatty acids. If it eats a low grain diet, it's going to be lower in certain types of fat. All grass, higher; all grass, lower. Does that make sense? Like you can actually manipulate the fat structure of animals. That's why that Kobe beef is so cool, because it's kind of like an engineering marvel where they found exactly what to feed the cattle in order to get this perfect marbling throughout it. So something that we kind of need to talk about is lipid in the diet and fat in the diet, and whether or not saturated fat is actually a devil or not. I'm going to sit here and tell you right now, it is not. Saturated fat has been demonized. There's nothing wrong with saturated fat in the diet. It is perfectly fine. If you look at the current evidence, it is super safe to consume saturated fat. Right? Super important to know that. This is kind of where we were talking a couple of weeks ago about how science can become wishy-washy. So this is a perfect example of it. It's the reason why like one day eggs are good and one day eggs are bad. Science can be entrenched. Saturated fat is a perfect example of time that diet nutrition research has become entrenched. So it's something that is less than ideal that happens, but it does happen. Now, am I sitting here and telling you to eat nothing but exclusively saturated fat? No. But what I want you guys to know is that it is not necessarily that dangerous for you. On the other side of things, fish oil is super important for you to consume, right? If you're not consuming fish in a somewhat regular manner, once or twice a week for this cold water fish, then you need to be considering supplementing with something like a fish oil supplement. And that's just simply in order to get all the essential fatty acids that you need inside of your diet. Another type of fat that we need to talk about is compound lipids. Two perfect examples of compound lipid. Lipid is a phospholipid and a lipoprotein. Phospholipids, think about the phospholipid bilayer. So it's actually a fatty acid molecule, two strings of fat with a phosphate group attached to it that creates kind of this polar being. And that polar being can actually create that cell membrane that is so important for humans to have. So it's really kind of a cool thing. Also, it's why dietary phosphate is super important and why we need to consume phosphate. Not to mention the fact we need phosphate for like adenosine triphosphate and stuff like that. Another form of compound lipid is going to be our lipoproteins. So that's our LDL and the HDL. These are super important. They're going to be formed in the liver. And what I want you to know is that all they represent is a transport mechanism for fat. It's how we're going to transport fatty acid, vitamins, fat-soluble vitamins, and it's just a transport mechanism for fat, right? If you need more fat to get to your muscles, you're going to have to increase things like LDL. It's just going to happen because you have to get that fat mobilized. So that kind of brings up this discussion about high density and low density lipoprotein. Think about what those words mean. High density means that it's going to sink. Low density means that it's going to float. It's why oil and water don't mix. It's why oil sits on top of water, because of the fat inside of the water. Our blood is predominantly water. So it's kind of where the nomenclature for these comes from. We actually get this by centrifuging it, and the HDL ends up pelleting at the bottom and the LDL ends up a little bit higher. In general, our low density lipoprotein are going to be fattier, more fat, less protein. So that's going to be our chylomicrons, which are ultra low density lipoproteins, which are almost exclusively triglyceride. Then we're going to have our very low density lipoprotein, or VLDL. It's going to have the highest amount of triglyceride of the three main lipoproteins of VLDL, LDL, and HDL. Then we're going to have our LDL, low density lipoprotein. So we're increasing in density, but it's still low density. These are going to be our bulk carriers of cholesterol. So that fat with a hydrocarbon ring. Now, cholesterol is not bad necessarily, right? We'll talk about it. It's not necessarily the worst thing in the world. And not all LDL is terrible for you. Again, stay tuned. HDL is high density lipoprotein. So this is going to have the lowest amount of cholesterol and the most amount of protein. If you were to drop a chicken breast in a tub, what's going to happen? The chicken's going to sink. But if you were to drop a drop of oil or a bottle of oil or 10 bottles of oil inside of a full bathtub, all that oil is going to float. So it's the higher the protein level, the more likely it is to sink. So the higher the density. If it's got high protein and low fat associated with it, it's going to actually be able to grab more cholesterol and kind of deal with that cholesterol that's in circulation. This is why HDL has the name good cholesterol, because it actually helps to remove cholesterol and help to excrete it via the reverse cholesterol transport mechanism. Let's kind of talk about the size and diameter and density of these. As you look on that far right-hand side, you can see that chylomicron clearly is going to be the lowest density. It's inverse, right? And it's going to be the biggest. It's going to be the fluffy. It's going to be full of the most fat. As we work our way down, you're going to see the diameter changes and density also changes. Now, when it comes to LDL, we're actually going to have different types of LDL. Not all LDL is the same. We're going to have large, fluffy LDL, and we're going to have small, dense LDL. That small, dense LDL is actually the dangerous one. The high density is not. So our derived lipids are going to be the ones that contain the hydrocarbon ring. Don't freak out about what the hell is a hydrocarbon ring. It's okay, right? Like I promise you, I'm not gonna kill you guys on this type of stuff right now. Essentially, derived lipids come in a couple different forms. Main one is going to be in cholesterol. And cholesterol is only in animal tissue. Like we're only going to get cholesterol in the diet from animal. You're not going to get it from plants. So it's going to be the most commonly found and known derived lipid. And it's going to be the most important inside of humans. This is the backbone essentially for all steroids. So whether this is bile, this is vitamin D, this is testosterone, this is whatever, a low-fat diet is painfully low in cholesterol, and a low-fat diet might compromise your actual cholesterol levels, which might compromise your hormone levels. So while it's not necessarily containing fatty acids, it's got similar characteristics, so we count it as a lipid. So something that's kind of recently been changing in the scientific literature has been the relationship between cholesterol and LDL and heart disease. What was previously thought that LDL was directly related with premature death, which means that as LDL goes up, you die faster, is not necessarily true. We've recently been seeing, and really with really strong science, we've been seeing recently that it's not necessarily all LDL that is bad. So we've largely grouped LDL into this one massive family and said that everyone in that family is bad. As you guys probably are well aware, anytime you segment out an entire group and say that it's bad, it's usually not the truth, right? Never is it the truth. So what we've really seen is that LDL is a family of species. So there's large fluffy LDL, which is big, kind of like less dense LDL, and there's small dense LDL. And that small dense LDL is the dangerous one. That small dense LDL is the one that can actually get in and cause some of this cardiovascular disease and heart disease. So when people talk about, oh, my LDL levels are here and I wish they were here, or the doctor said I need to lower my LDL, something that you guys need to understand is that not all LDL is created the same. And more often than not, when someone gets a blood report back, whether it's your parents or yourself, that has an LDL number on it, that is not actually a directly measured LDL. Almost never is it actually directly measured. So it is completely an estimate. Right? That's kind of scary that we base a lot of clinical decisions off of a guess, off of an estimate. When it is possible to measure it, it just costs a little bit more money. So just ask. Next time you hear someone talk about their LDL levels being super high, simply ask and say, hey, like, have you ever had a particle count done for your LDL, or have you ever done a size test for your LDL? And you'll be shocked to hear what they say, because it's probably going to be no. So what that kind of means is that we have two different patterns of LDL that really start to present itself. We have a pattern A and a pattern B. That pattern A LDL is going to be the large, fluffy, and benign LDL for the most part, right? This is going to be the one that's not really causing any disease whatsoever. It might actually be protective in many ways. Our pattern B LDL is going to be our small, dense, high-risk LDL. This is going to be the one that's more likely to cause damage, more likely to get in between the intima of the vasculature and cause these fatty acid streaks and plaques and stuff like that. So what do we know about LDL? We know that it's not necessarily as bad as we once thought it was. We know that there's a couple different patterns of it, and we know that one pattern is worse than the other. We know that pattern B is worse than pattern A, and that pattern A LDL is not necessarily a bad thing. Right? Super important. Trying to get you guys to think a little bit more advanced, a little bit more complex here. Something else that I want you guys to kind of think about is that there's no daily recommended intake for fat. if you really truly read what the dietary guidelines say, and you really truly read what some of these other groups are saying, there's no high level of fat intake. They put recommendations out there. Yes, 100%. There's recommendations out there for the amount of fat that should be consumed in the diet. But there's no hard data that shows what a high-fat diet is necessarily going to do. We know what a low-fat diet is going to do. We know that there's actually really bad health outcomes when you have too low of a fat of diet. So what you'll see instead is that people actually start putting recommendations on cholesterol instead of on fat. Like, you should limit cholesterol to 300 milligrams per day or less. It's not a ubiquitous approach, right? Like, this is something that should be, for most of you guys, setting off kind of skepticism, light bulbs, if you will, where we have really firm recommendations for carbohydrates despite the fact that it's not needed whatsoever in the diet. And when it comes to fat, there's really not a whole bunch of recommendations against it, but we almost never consume it, or we're not recommended to consume it. It's a really weird phenomenon. So am I telling you that you need to eat as much fat as possible? You need to recommend someone always eat as much fat as possible? No, that's not what I'm saying. What I'm saying is that use science, start to look at it, and try not to demonize food groups. Just because fat is fat, don't demonize it. Just because carb is carb, don't demonize it, right? Each one of those things has an importance inside the body, whether it's derived, whether we're creating it, or whether we're consuming it. Just kind of think through some of these things. With that being said, why do we actually have lipid inside of the body? We've talked about what it does. We've kind of talked about some of it, like what kind of some of the structure it ends up as. But why do we consume it? Well, we need lipid in the diet because we need energy. Those of you guys that have had phys before, exercise physiology, know that bioenergetics, we actually can create ATP off of lipid. For every 1 gram of fat, we actually get 9 calories worth of energy. kcal is the same thing as a calorie. It's kind of a unit of measure. So we know that we can actually burn fat and get energy from it. We also need it for protection. Think about if you've ever been punched in the stomach, sometimes you might wish you have had more fat there to kind of absorb the blow. We're actually trying to protect our internal organs by wrapping them in fat. We use it as insulation, so when times are a little bit cold, we use it to kind of keep heat inside of our body. And we use it to transport fat-soluble vitamins. Again, just because blood has fat in it does not mean it's a bad thing. We have to transport vitamins and we have to transport energy. And what you'll see in the hunger, what you'll see in the digestion section is that there's actually hormonal control to actually control hunger. So we can significantly depress hunger by consuming fat and significantly depress the feeling that you need more food by eating fat. Kind of a really cool substrate in the body. I just want you guys, I don't need you guys to worry about having to draw these or whatever. I just want you to be able to visualize what they look like. So cholesterol, you can see you've got all these kind of hydrocarbon rings. That's why each one of these is kind of circular-ish, hexagonal in structure. I guess these are 1, 2, 3, 4, 5. Yeah, it's hexagons. I'm an idiot. I thought it was an octagon for a second. And you can see that this is a free fatty acid, right? Just like we were saying before, it's actually an omega 1, 2, 3, 4, 5, 6, 7, 8, omega 9 fatty acid. Here you have a triglyceride that's attached, so it's three fatty acids attached to a glycerol backbone. And here we're going to have a phospholipid, so we're going to have 2 fatty acid chains, a phosphate group, and you actually have a nitrogen associated with it as well. So kind of cool, right? We've covered fats and we've covered carbs. You should be kind of excited. Plus, you got only 9 slides left, and you're done. So let's get a move on. All right. So, nature of protein, or should I say gains, right? I don't know. I'm an idiot. So, realistically, the major difference between carbs and fat is what? It's the carbohydrate to hydrogen. It's the carbon to hydrogen ratio, right? Major difference in carbs to fat, carbon to hydrogen ratio. When it comes to proteins, major difference is going to be the presence of an amino group or the presence of that nitrogen group, right? So if you look over here, you can see that left-hand side, you're going to have nitrogen. This R side chain, that's going to be what makes the individual amino acid the individual amino acid. It's what gives it its unique character. Every amino acid is going to have a separate and a different side chain. So it creates a uniqueness to it. It's its haircut, right? It gives it a uniqueness. Amino acids are just the building blocks for proteins. Individual amino acids end up combining together to create a protein. They're joined by peptide bonds. So amino acids are actually joined by peptide bonds. So if we have 2 of them together, it's called a dipeptide. 3 of them together, tripeptide. Anytime you have more than 100 of them combined together, that's when we get a protein, right? So kind of cool. It's building blocks. It's Legos, if you will. Put a whole bunch of different Legos together to get a really sweet, like, laser blaster or something. I don't know what Legos do anymore. As far as kinds of protein, we're going to have a couple different types of amino acids, right? So we're going to have essential and non-essential amino acids. Just like we had essential fats, we're going to have essential amino acids. This means we cannot make them in our body. We have to consume them. There's certain conditionally non-essential amino acids, but don't get caught up on that right a second. When it comes to essential, we have isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Non-essential amino acids means that we don't necessarily have to consume them because we can make them in the body. We're also going to have conditionally non-essential amino acids, which means that as long as the conditions are right, as long as we have the correct precursor amino acids, then we can actually make them as well. In general, our non-essential amino acids, conditional or non-conditional, are going to be alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine, right? A couple of them should kind of, if you're a sports supplement guy or female, should kind of stick out to you as branch chain amino acids, and that's leucine, isoleucine, and valine are our essential amino acids. Those are going to be in those BCAA supplements that you're taking. Now, animals and plants both produce amino acids. You can get a full complement of amino acids from consuming plant sources. You don't have to consume meat in order to get all of your amino acids. But the amount of complete protein in plants compared to animals is significantly less. So you have, like, if you want to not eat meat, but you still need to get your protein, you have to actually start combining carbohydrate and plant sources in order to get the complete ratios. A complete protein is what you're going to see in animal products and meat-based products, and that means that it's containing all of the essential amino acids in the correct ratio as well as the quantity. So incomplete proteins, plant proteins are not. Animal proteins, complete proteins are. So what are some protein sources? Well, animal. Anytime you're eating meat, you're eating muscle. Anytime you're eating muscle, you're getting all of that protein that you need. Realistically, most people get about 2/3 of their protein from animal sources. That could be eating chicken, steak, whatever. Me personally, I'm a venison and a red meat type of person. I prefer to eat red meat over chicken just because I like the way it tastes, and because I like it. That's really the only way. But some people don't like meat or would prefer to not consume any animal-based products, so they go for vegetable sources of protein. No matter what you do, you have to consume protein. If you do not consume protein, you will waste away and you will die. Our body has to have it. It is requisite. Just like fats are requisite, protein is requisite. Unlike carbs, which are not requisite, right? So when it comes to complete vegetable protein combinations, it tends to be like a legume or a nut and a seed of some way, shape, or form. So we can start having rice and beans and stuff like that in order to get whole grains, in order to get whole proteins. Sorry, I read whole grains over there on the right-hand side. So it's the combination, different amino acid combination groups from different plant sources in order to complete a full complement or complete protein. Now, the RDA for protein is actually set relatively cleanly, right? The current RDA for protein is 0.8 grams per kilogram, or about a gram per a gram per half pound-ish, because 1 kilogram is 2.2 pounds. Regardless, the current RDA is 0.8 grams per kilogram. It ends up being a decent amount of protein, and what's nice is that it's a threshold that's set a little bit high intentionally. So that is the value for the minimum amount of protein needed in order to not just lose protein, lose muscle all the time. Now, if you're more than active, if you're more than the sedentary individual, if you like to weight train, if you're an athlete, if you complete manual labor of any way, shape, or form, you're probably going to need more than the RDA of protein. The current athletic guidelines scale all the way up to about 1.8 to 2 grams of protein per kilogram of body weight, which ends up getting pretty high. You'll oftentimes hear someone argue that, oh, that much protein is bad for you. It's not. Research has gone all the way up to 4.4 grams per kilogram of body weight, which is a ton of protein with no harmful damages. Protein's not going to kill you. The big thing here is just making sure that an individual has completely healthy and functioning kidneys. If an individual is on dialysis or they're on some sort of kidney disease protocol, then high protein might not be the best thing for them. But in general, it is a very safe macronutrient to consume in high levels. So what happens if we really fail to get that protein? If we fail to bring that protein in the diet? Well, the big thing is our body needs it. So our body's going to get it. It's going to start sapping our own muscle stores, pulling everything from our own stores in order to get the protein that it needs. So we will break down our own skeletal muscle. We will break down our own cardiac tissue if we need that much protein, right? Like we will lose muscle mass if we need protein. So it's super important to get protein in the diet. So that's not just a performance loss and a loss of some peaked biceps, but it's also a loss in health quality as well. So what does our protein do? So many things, right? It is one of the key structural components in our body. Massively important when it comes to structure in the body. It's 15% of our body mass. Think about that. That is insane. And if you go with that 15% of our body mass from one macronutrient essentially, then we've got 65% of that is coming from muscle protein. So our muscle weight is huge here, right? How much muscle we have is going to be driven in some way, shape, or form by our protein quantity in our diet, as well as our hormones and stuff like that, right? There are also amino acids. Individual amino acids play a massive role when it comes to DNA and RNA synthesis. They're actually the backbone. Amino acids are the backbone for DNA nucleotides. So our genes, our genome is basically based in some way, shape, or form off of amino acids. Also play a role in like hair and skin and bone quality and stuff like that. But in general, I mean, it's just a super, super important macronutrient for us. When we go to actually metabolize or break down protein, what we'll talk about in digestion and absorption is that the breakdown or catabolism of amino acids is essentially getting a long protein, which is 100 plus amino acids, down into individual amino acids and then deaminating them. So actually pulling the nitrogen off of them. So what happens with protein metabolism is we get a protein that's hundreds of amino acids long, break it down into individual peptides, break that down into individual amino acids, and then we pull the nitrogen off of it, and that's called deamination. And then we have to excrete that nitrogen in some way, so we actually help to create urea and then urinate out that urea so that we can eliminate that free nitrogen and kind of help eliminate the nitrogen products inside of our body. So that leads us to this concept of nitrogen balance, right? If on one side we know we're excreting it and on one side we know that we're eating it, we kind of have to start weighing this balance of intake versus output, intake versus use, right? Or intake versus excretion. And so when we're in a positive nitrogen balance, it means that we are consuming protein at a rate that is at or above the needs within the body. So we're actually going to be in what's called an anabolic state. We're going to be in a building state. We're not going to be breaking down our own body's amino acids because it's going to be getting the protein that is requisite within it. So nitrogen balance is about simply making sure that we're consuming enough protein to at least match, if not exceed, the body's breakdown. If we're in a negative nitrogen balance, that means breakdown or catabolism is exceeding consumption or anabolism, which actually means that we're actively losing skeletal muscle and we are actively breaking down our own protein stores because we're not getting enough of it. So nitrogen balance for the athlete, incredibly important. Nitrogen balance for the patient that is like bedridden, incredibly important. Think about like burn victims that are bedridden. Think about AIDS patients who are in a kind of this constant state of inflammation and protein breakdown and skeletal muscle wasting. Nitrogen and protein is incredibly important in these individuals. And then lastly, I just wanted to kind of highlight one kind of cool cycle that you can do with amino acids, and we can actually literally take and make glucose from amino acids. So if we start here in the center, you can see that we have a muscle protein that gets broken down into individual amino acids. We deaminate it, we lose that nitrogen, we lose that NH4, and we end up with a glutamate. We use this liver enzyme, ALT, that frequently gets measured in blood labs, and we create an alpha-ketoglutarate and an alanine. That alanine then kind of moves over to the liver, gets shot out of the muscle into the blood, transported into the liver, and then eventually we create glucose all the way down from it. Kind of a really neat situation that we can do to actually use muscle for energy if we need it, if we're in the wrong energy state. All right, gang, that's it. Video 1 of the 3 that I owe ya. So get a move on, and hopefully this helps. I know that most of you guys, actually only a couple of you guys asked for this, but please use it. It's here. Use it. I'm sure that you guys will get a lot of use out of watching these videos.